Hi, my name's Joe Anderson. I'm an associate professor at the University of California, Davis. I'm in the Department of Internal Medicine, Division of Infectious Disease. I was always interested in vaccine development, therapeutic development, how we're able to sort of manipulate viruses to use for our own advantage. When I started my PhD, I got into a lab that worked on HIV and gene therapy, and that was my first introduction into gene therapy. And then we also worked on stem cells, the stem cells that make the immune system and all the red blood cells in our cells. I then wanted to use sort of similar techniques, the vector system that we use, the stem cells that we use, but use the strategy for other diseases and other disorders. So we moved on to some neurodevelopmental disorders, including Angelman syndrome. And once we saw that it worked well on Angelman syndrome, we're now trying to apply it to other neurodevelopmental disorders, including Rett syndrome. We would be taking a patient's own blood-forming stem cells. It's in the bone marrow of everyone. That's what constantly makes our red blood cells and our white blood cells and our immune system cells for the rest of our lives. And we would be genetically modifying those stem cells with a functional copy of the gene so that once those stem cells are transplanted back into the body, they'll engraft back into the bone marrow, and then they will make protein and enzyme expressing cells throughout the body, including in the central nervous system and in the brain, for the rest of the life of the patient. And so we target the stem cells because they're there for the rest of the life of the patient, so it could potentially be a one-time treatment. So the procedure would be similar to a bone marrow transplant that cancer patients receive. And so the bone marrow shouldn't be affected by Rett syndrome, and so we would be able to take a patient's own bone marrow stem cells. And then what we do in the manufacturing is we purify these stem cells from the blood that we receive from the patient. We genetically modify them with our vector system. And then once the patient is ready, then they receive the therapeutic cells in the vein. So it's an IV infusion. The nice thing about these stem cells is that they know where to home. They know to go back to the bone marrow. That's where they live. So you just need a IV infusion of the gene-modified cells, the therapeutic cells, and then they go back into the bone marrow. And then over time, we would take blood samples from the patient, and we would look and see how well are their blood cells marked with this therapeutic gene, with the therapeutic protein. We would look at expression of the protein. And then we would also look at, clinical phenotypes. Are they improving? So the first steps that we need to take is to see if it will actually work in preventing and or rescuing the phenotypes, so the efficacy or the effectiveness of the therapy. And once we show that that works, then we do toxicity studies in another animal model, because we are overexpressing this therapeutic protein, and it will be expressed constitutively, which means all the time, in these immune cells in the central nervous system for the life of the patient. So we have to do these toxicity studies in an animal model to show that overexpression of this protein doesn't cause any harm. The good thing is that we've done this in the past. We're currently doing it now for our Angelman syndrome work, so we know what the FDA asks for to be able to start a clinical trial. And so we know the path that we have to take. We just have to first show if it's effective and then show that it's safe. It's great when you get connected with a foundation that, is looking for high risk, high reward sort of therapies and therapeutic strategies. When there's no approved therapy or therapy that has proven to work really well for any type of disorder, it's great when funding agencies are able to fund and want to fund all different avenues, because you never know what's going to work. And so it's great when you just put all hands out for any type of therapy that potentially could work for any disorder. Finding the Rett Syndrome Research Trust, and they loved the proposal that we have. They see how well it worked for Angelman syndrome. There's a good possibility that it will work as well for Rett syndrome. And yeah, we're excited to start it. Excited to put it in the animal model and see how it works and move forward from there.